Greetings, Pencrest biology students. This is Mr. Del Prado, and the purpose of this video is to go through our whole DNA unit from beginning to end. Um, Purpose of this is maybe for students who missed a couple classes and want to get caught up on the basic tenets of DNA, or for students who just wanted to do a little review. This unit can get a little vocabulary intense. So I'll start off with the structure of DNA and I'll go all the way to the end with translation. Okay, so remember that your DNA is different from everybody else's DNA on Earth. It is that variability in DNA that makes us all different from one another. Not only one human to another, but a human from a maple tree or a rhinoceros or an elephant. Those differences in DNA account for our physical and biochemical differences. So, central dogma in biology, or one of the central themes in biology, is that DNA is a nucleic acid that provides a code that tells our body which proteins to make. That DNA is going to get converted into RNA, then that RNA is going to get translated into a protein. Okay, we went over this a fair amount in class. I think hopefully you guys got something out of the story behind this. I won't go through that now. Just recognize that Watson and Crick are generally the two scientists credited with discovering the structure of DNA. But remember that this lady was one of the linchpins to make sure that they were able to discover that structure. Rosalind Franklin's the one who took the X-ray picture of DNA that you can see there that Watson and Crick figured out, oh, it's a double helix, it's a twisted strand, and that is the structure of DNA. Okay, so Rosalind Franklin is somebody that you should know. Watson and Crick is somebody that are people that you should know. So DNA, the shape, it's a double helix. It consists of nucleotides, and this is the first vocabulary word where things get a little confusing. A nucleotide is just a short piece of DNA. Scientists will call this a monomer, and if you look at a nucleotide, you can see it's essentially based on three components, and here it is right here. There's a phosphate group in DNA. There's the sugar in DNA, which is called deoxyribose. And then there's one of these four bases, adenine, thymine, guanine, or cytosine. Okay, those are the four bases that essentially provide the code for DNA. Those bases are arranged in a very specific order in that adenine always bonds to thymine and guanine always bonds to cytosine. Is that 100% true? No, but we'll get into genetic mutations a little bit later in the year when we talk genetics. But for the most part, A bonds to T, G bonds to C. Those bonds, if you will, aren't really bonds. They're attractions. They are hydrogen bonds that connect the bases together. So sugar phosphate backbone, okay? And then the bases are going to be in the middle. All right, there's a picture of DNA. The picture over on the right is showing a little bit more detail. It's showing the phosphate and sugar groups. The picture on the left there is kind of ignoring the sugar phosphate backbone and is looking, where the DNA is. Remember that DNA has a starting point and an ending point. If your starting point is a phosphate coming off of carbon number 5, we call that the 5' end of DNA. If your starting point is a hydroxide coming off of carbon number 3, we call that the 3' end of DNA. A great way to think about it is it's said to be anti-parallel, but a good way to think about it is it's like cars on a highway. They're going parallel to one another, but they're going in opposite directions. Okay, so DNA is a nucleic acid. It's a double helix, A to T, G to C. All right, this is where it starts to get interesting. So your cells have to grow, they're going to replicate, and then they're going to die. All right. So this process of replication ensures that when a cell, before a cell dies, it actually replicates itself. So there's backup cells, so you don't just turn into, a puddle of mud. All right. So as your cells age, they're going to replicate, and then they're going to die. This process, DNA replication, is just explaining how the cells replicate their DNA before a cell actually divides. So if you look at this picture here, you can see a picture of DNA. And again, I'm going back to this 5' to 3' direction. If you look at the sugar phosphate backbone on the left, you can see it's running in the 5' to 3' direction from the top of your screen to the bottom of your screen. And then if you look at the sugar on the right, it's going 3' to 5' from the top of your screen to the bottom of your screen. Kind of important because DNA only gets made in the 5' to 3' position. or 5 prime to 3 prime position. What do I mean by that? Well, let's look at the process. Okay, the first thing that's going to happen is helicase is going to unzip the DNA strand. Okay, it breaks those hydrogen bonds. Then bases are going to be added to each side of the DNA strand, and you can see that in your picture here. The bases A to T, G to C are going to ensure that that top strand and the bottom strand are exactly the same. The big kicker to this is that DNA polymerase, the enzyme that adds the bases to the DNA molecule, only makes DNA in the 5 prime to 3 prime direction. What that means then is if you look at our top strand over there, the DNA is unwinding and that DNA polymerase is moving towards the replication fork where the DNA is being separated by the enzyme helicase. Okay? It is making DNA in the 5 prime to 3 prime position, essentially moving as soon as the replication fork opens up. But on the bottom strand, DNA polymerase, it also has to work in the 5 prime to 3 prime direction. But you will see the enzyme on the bottom, DNA polymerase, is working away from the replication fork. So it has to make little pieces of DNA at a time. Those pieces are called Okazaki fragments, and then they are actually sealed together by the enzyme DNA ligase. The end result, though, is going to be essentially that you're going to have 2 strands with exactly the same DNA. I put this picture in here just to give you a little clearer representation of what's going on. Excuse me, with replication. You can see that blue triangle is helicase unzipping the DNA strand. You can see DNA polymerase. It's orange on the top strand. It looks yellow to me on the bottom strand. Okay, on the top strand, it's making it in pieces. That's how you get those Okazaki fragments. And DNA ligase is going to seal them together. And on the bottom strand, it's DNA polymerase making it in one continuous strand. A couple other players that you would need to know for DNA replication. One is this green enzyme right here. That's topoisomerase, and that enzyme flattens out the DNA so that it can be split apart. You should know that DNA primase is an enzyme that lays down an RNA primer that tells DNA polymerase where to start adding the bases for DNA. DNA ligase seals it on up. The Okazaki fragments are only found on the lagging strand where DNA polymerase is moving away from the replication fork, which is away from the enzyme helicase. Okay, for my visual learners out there, hopefully this little animation will help you out a little bit. You can see DNA polymerase, it's that pink enzyme. Don't worry about DNA polymerase 3 or DNA polymerase 1. Okay, it's DNA polymerase, and you can see on the top strand there, it's moving towards the replication fork. That's our leading strand. On the bottom strand, DNA polymerase is making DNA, but it's moving away from the replication fork. So it's making the DNA in little tiny pieces. Those pieces are the Okazaki fragments. I am presuming at some point here in just a second, we're going to see a DNA ligase that's going to come in, and it's going to seal up those little fragments, which are the fragments. But again, top strand and the bottom strand would be exactly the same. Okay, this replication, it is occurring at breakneck speed. There are 3 billion bases in just one of your cells, so it takes about 4 hours for DNA to actually replicate itself. That little thing that I have down there about sometimes replication errors occur, it's true, and we're going to get into more depth in that a little bit later this year. Another important vocabulary term that you should know is that if you look at those 2 DNA strands, they're clones of one another. But part of the new strand actually has the old strand in it. You can see that the yellow is the new strand, but that blue is the old strand. That's called semi-conservative replication. And all that means is that even though we have 2 new strands, part of the new strand is actually made up of the old strand. This is a computer simulation of replication that is actually going through in your cells. You can see helicase is going to be right here, unwinding the DNA strand. Okay? And then you should be able to see there's going to be some DNA polymerase that's going to make new DNA. This is going to be our leading strand. See how it's working continuously. And this is going to be our lagging strand that's going to make it that DNA in little pieces. So that's DNA replication. We're making exact copies of DNA before the cell actually divides. Separate from replication is the process of making proteins. And for that process to occur, we need to look at 2 features of DNA inside the cell. So here's the big picture. DNA is going to get made into RNA, The RNA is going to get made into a protein. Just to be clear, this is not part of replication. It is completely separate from replication. Don't get caught saying that the cell goes from replication into transcription. They're two separate processes that occur in the cell, but we talk about them together because the processes are similar to one another. Inside your cells, you have long segments of DNA, 3 billion base pairs long, A's to T's, C's to G's. The DNA is separated into segments, and some of these segments are small, some of the segments are big. Those segments are called genes. Those genes are sequences of DNA that actually make proteins. Okay, so going from smallest to biggest here then, you would have an atom. An atom makes up a nucleotide. The nucleotide makes up the genes. Genes are going to make up those proteins, and then a bunch of proteins are actually going to make a cell. All right, so here we go. In order to make a protein, our first problem is that DNA is too big to leave the nucleus. Some of you may remember me talking about that in class, I hope. Okay, so DNA gets made into something smaller and more stable. It gets made into RNA. That RNA essentially takes copies of the DNA, and it's going to take it out of the nucleus. DNA is trapped inside the nucleus, so it sends its message, RNA, out to the ribosome. Okay? RNA is a nucleic acid, very similar to DNA, but it's single-stranded. It doesn't have the base thymine. It has uracil, so that means A is going to bond to U. It's going to have the sugar ribose, and also, too, it can be found in the nucleus, but because it's so small, it can go through the nuclear pores as well. Okay? I feel like I just went through the basic differences between DNA and RNA, and that's kind of a big thing for you to know in class. How do we turn DNA into RNA? Okay, we use a process called transcription. Transcription is similar to replication, but not exactly the same. Okay, take a look at the big pink enzyme there. That pink enzyme is RNA polymerase. It does two things in transcription. It unzips the DNA strand, and it adds the RNA bases to the DNA strand. The kicker is this: it only transcribes in the 5' to 3' direction, and only 1/2 of the DNA strand gets transcribed. Okay, because we're going to make this into a smaller message. So RNA that's going to be made by that RNA polymerase is going to be single-stranded. RNA polymerase is going to add the bases in on one side of the strand, and our end result then is going to be that you're going to get a single-stranded RNA molecule. So I'll give you an example. Just looking at the first line up there, if that's our DNA code, if we were to take RNA polymerase and make the code from that, the messenger RNA, we're taking the message from the nucleus out to the ribosome, the sequence of bases would go, I'm looking at the top line there, U, U, A, C, G, U, A, C, G. You can do that on the bottom two strands on your own. But we're going to take DNA, we transcribe one side of the DNA so we get a single-stranded messenger RNA. You can see the picture. There's our RNA polymerase. You can see our single-stranded messenger RNA that is going to be small enough now to leave the nucleus and go to the ribosome. Before it does, there are a specific set of enzymes that cut out junk DNA. They're called ribozymes. And what they do is they cut out junk DNA. Junk DNA is also known as an intron. So it cuts those out, and then you're left with exons, which are the portions of DNA, now RNA, that are then going to be used to make a protein. All right, so now our messenger RNA has left the nucleus. It's going to go to the ribosome. The reason we call it translation is because we're taking a code, A's, U's, C's, and G's, and we're actually going to make proteins out of it. How does this actually work? Well, if you take a look at our bottom right there, again, for my visual learners out there, you can see that pink acorn-looking thing. That's the ribosome. You might remember they're all over the cell. They're on the rough endoplasmic reticulum as well. Okay. On the bottom, you can see the single-stranded messenger RNA. I think it's red in color. It looks like the single-stranded. It's on the bottom of the ribosome. Another player comes in. It's called transfer RNA, and it's going to bond to the messenger RNA. Each 3 bases on messenger RNA are called codons. The bases that bond to them found on the transfer RNA, those little crosses, they're called anticodons. But transfer RNA doesn't just bring anticodons. Look at those little circles on the top of the transfer RNA. They also bring amino acids. Those amino acids connected together